all the esteemed researchers and the audience members who are joining this event. I am Manisha Kalsan, a postdoctoral research scholar at Jawaharlal Nehru University, New Delhi, India. Today I am going to moderate this session. The AP Bionet, founded in 1998, promotes bioinformatics in the Asia Pacific through collaboration, networking, and training programs. Is there an issue?
Yes, you are
Partners?
audible.
It partners with the Asia-Pacific Advanced Network to enhance infrastructure and collaborates with organizations like DDBJ and GenomeNet for data exchange. It has multiple initiatives such as the Bioinformatics Research Consortium, which unites experts to tackle complex research challenges. The Bioinformatics Grand Challenge Consortium addresses global issues, while the Student Ambassador Program fosters skills and international collaborations among students in the region. The Bionet sponsors workshops, symposia, and conferences to support the community. Today, we are gathered here as a part of its initiative, AP Bionet Talks, which serves as a platform for bioinformatics education, sharing the latest developments and training. Today, we have as our expert Dr. Luis Pedro Coelho. He is a group leader at the Center for Microbiome Research at the Queensland University of Technology. His research focuses on using very large-scale datasets of the global microbiome to understand microbial ecology. His group is also known for developing high-quality tools, most notably SemiBin for metagenomics winning. Before moving to Australia, Louis got a PhD from Carnegie Mellon University in the US, worked at the EMBL in Germany, and at Fudan University in China. Today he is going to give a talk with the title Finding Antimicrobial Peptides in the Global Microbiome Using Machine Learning. So antimicrobial peptides are small peptides which kill or inhibit microbes. They are produced by organisms from all domains of life and have been a topic of interest for drug development. Dr. Coelho and his team has worked on weaponizing these peptides, and it will prove to be a wonderful step forward in the field of drug development. So let us all welcome Dr. Coelho.
Thank you. Thank you so much for that introduction, and also thank you for everybody from being here. So I will start sharing my screen now. And okay, you should see my slides now. And I'm going to post in chat. So if you or I just posted in chat, so the slides are publicly available at this URL. So the slides. So all right, so without further ado, I'll just go ahead and get started. So yeah, so my name is Louis, and I did have a couple of slides just on how I got here. Oh, sorry, I made a mistake there. I just posted in chat the slides, and I I started my research and academic career in Lisbon. I did a bachelor's and master's in computer science, but then during that time I started to get a bit of biology FOMO, and I thought, okay, biology is really I thought at the time is a bit more interesting than what I'm doing, so I'm going to start doing a little bit more computational biology, and then I went to Carnegie Mellon to study computational biology. Although at the time it was a relatively different project than what I work on now. I was doing a lot of more eukaryotic cell biology. Then I switched into the field I'm now in metagenomics because I was in Heidelberg at the EMBL, and I did a postdoc with Pedro, who's a really important researcher in the field of microbiome, and that's when I started working in this area. So in 2018, so I started my own research group in Shanghai at Fudan University as a junior principal investigator, and I was there until about a year ago. So it was in September of 2023 that I then moved here to Brisbane to QUT. So I've been here for a year, but I was already in the Asia Pacific region in Shanghai before. I know, and then today I'm here, sitting in my office, but I'm presenting to all of you. So thank you for being here. So I want to go back a couple of years, not too much, to talk. So this was work that I was doing before, where we were looking at the global microbiome. So when we use the term global microbiome, we mean that, so we'll try to look at the microbiome of the whole planet, all together, and try to understand what's going on from the perspective that all of these things are linked. So all of the work I'm going to be showing today is based on using publicly available data. A very tiny amount of this data was data that we collected ourselves, but 99% of it is actually data that people collected and they put it in public databases, and then we are doing a reanalysis of it. So a lot of these, so in these manuscripts, they don't actually have new metagenomes. So at the time, we had this data set of over 10,000 metagenomes. I mean, in the meanwhile, the field really has, it grows exponentially. So every year, basically every year the field almost doubles in the amount of data that's publicly available. But so even a few years ago, 10,000 was a very large number, and now we're talking about hundreds of thousands. And we looked at this, and we had these different. So here I've colored them by color. So human gut, mouse gut, cats, dogs, other human associated environments. We had some sort of environmental, marine, and soil, and this is, and you can see if you looked at, there's the we say global, but at the time this was very, very poorly sampled. So there's basically no dots in the continent of Africa, and there's basically nothing in South America either. So, but we took what we had available then, and what we do is, so we take these metagenomes and we predict genes from them. So we predict protein coding genes, so open reading frames. And because people will sequence the same organism multiple times, that means that we will get the same sequence multiple times, right? We will, and so we dereplicate this. So we start with, we have 2 billion of these potential genes, potential open reading frames, and then so we also took genes that came from Progenomes 2, which is a database of genomes. So these are cultured, not cultured species. And we first dereplicated them, and then we dereplicate. Here, this means that we got exactly the same sequence, so exactly the same nucleotide sequence. And now we're down to a billion. But it's still, having just the exact same sequence, this is a little bit too strict. So we further cluster them into different groupings. And the most important groupings are these groupings that we call unigenes, which correspond more or less to the level of a species. So in the bacterium, a species is 95% nucleotide identity. That's it's not the perfect threshold, but that's a pretty good rule of thumb. I mean, this isn't like for example, in humans. So humans, we are all 99.9% identical. We are all almost. But bacteria, they're much more diverse in terms of their genomes, and so 95% nucleotide clustering corresponds to a species level, and then we have a couple of intermediate levels, but then we also have this higher level of protein families, which corresponds to basically any 2 sequences that share a common ancestor. So even if that this can be very distant, and so it can mean that nowadays their identity is very, very low. Maybe I'll just mention one result that we had from the time, which was that we saw that when we put everything together, in these plots that I'm showing you now, so the width of these ribbons represents visually how much sharing there is between the different habitats. So and if you look on the left, these are the broader groupings. So there's not that much sharing. There is still a little bit of sharing between, say the mammalian guts and other mammalian body sites. There's some sharing between the built environment and some of, because the built environment is often just a function of the people who are there. But if we look now on the left, and I'm just going to, where we have the mammalian guts, we can see that there's a lot more sharing. So, and actually, so we had previously published that the dog gut shares the most with humans, but actually, as it turns out, it's actually cats share even more, just slightly more. But it turns out that it seems that it's the pets that live with us, and not the pigs, and not the mice, which we use in experiments often. But it's the pets that live with us that end up sharing a lot of our microbiome. So this is what we were doing at the time. We published this already. So this came out in 2022, and so in science always asks, okay, so what's the next step? And on the one hand, the next step for us was to go larger. And going larger means, we want to see more. As I said, sequencing keeps increasing, and so we took another snapshot of what was available, and now we went from 10,000 to about 60,000 of these metagenomes. And you can see that, first of all, there's a lot more dots. There's a lot more dots. So whereas there's a lot more dots now also in the continents of Africa, there's a lot more dots in South America. Still not, not a good representation, but it's a little bit less unbalanced, so to say. And we also have a lot more different habitats. So here, just because I cannot display 70 different colors, I've grouped them into human gut, the soil, other humans and mammalians and other animals. But we have a lot. So there's been a lot more exploration of these, and we've also published them. And if you follow the link for if you follow the link, these are links. So we've published this as a database called Spire. So this came out earlier in the year. But we also wanted to go smaller, because this is the main topic of the day, is that one thing this information was always there. We were never hiding it, but it was always somewhere in the methods that we would say, oh yes, we were looking at all the ARFs, but the ones that are too small, we are going to ignore, because they're just difficult. And basically everybody in the field, with an honorable exception of Amibat's group, this is what we were doing a few years ago. So we were not looking at these small proteins, and so small proteins, they're not just smaller, so everything becomes much more difficult. They're much more difficult to predict. And some of this just comes down to you don't have that many amino acids to work with, and a lot of the statistical arguments just break down. So if you have a big stretch of DNA, and you have a start codon, and then you have hundreds of codons, and then a stop codon, this by itself is already evidence that there's been some selection going on here, because that doesn't happen by chance. By chance, sooner or later, you'll have a stop codon somewhere. And so if you see the absence of a stop codon in a long stretch of DNA, that by itself is already, you can already take that as some evidence that this is a something there. Right now, there's complications, but this completely breaks down if you only have 30 amino acids. You can have a start codon and then 30 amino acids, and that can be completely random chance. In a genome, a bacterial genome, you're going to expect that you have these stretches of start to stop of 30, 40, 50, up to 100 amino acids that are just completely meaningless. There's not just from that alone, you cannot infer that selection has been going on. And the same sort of logic also applies when you have homology. So often, a lot of what we do is based on homology, and homology is often, we bootstrap on sequence identity. But again, if you have very few amino acids, sequence identity becomes really difficult, because again, if you have hundreds of amino acids, you have two sequences with several hundred amino acids, and they are, say, 50% identical. This does not happen by chance, right? The probability of this happening by chance is infinitesimal. And so you can take this as pretty strong evidence that these two sequences share some sort of ancestry. And so you can take this as pretty strong evidence of homology. But if you have two sequences that are 40 amino acids and 20 of them are the same, 20 amino acids is just not enough for you to make any conclusion. So when I was starting my own group, and Célio Dias Jr. Was the first postdoctoral researcher to join my group, so he was very interested in this question. And then Yiqian Duan, who's a PhD student, still in my group. They were the people driving most of the work I'm going to show today. Because what we did is we took a little bit the same approach that we did before, where of taking everything together. And so as I said, there's about 60,000 metagenomes. Again, we put some of the isolate metagenomes in the pot, and we predicted this small open reading frame. So again, we're predicting sequences up to 100 amino acids. And we collapse the redundancy. So if we get the same sequence twice, there's a little bit of a complication here that I'm not going to go into, but we only take sequences that we see at least 2 times. So we ignore singleton sequences because again, they might, if we only see a sequence 1 time, that might not be enough evidence. But if we see it 2 times or more, then we start to think that that's something real. And then Yiqian also developed a tool that you can use. So if you have your own sequences, again, maybe in your data set you only see them once, but if it turns out that in our data set it's seen multiple times, maybe you can use that to build your own confidence in your own data, right? So, but if there's one thing that we saw from this survey is that we see these sequences, and here, so these are these identifiers, these numbers, they correspond to some set of sequences. So like, for example, this first one, 287.924.202. So this is present in Actinomyceta, it's present in Dormibacterium, so it's present in Sumanodota. So it's present in all of these different phyla. And here, so it's conserved evolutionarily across vast regions. And here on the right, we also see that it's present in multiple habitats. So it's also prevalent from the point of view of ecology. So we have these sequences, they are present everywhere. They are present in multiple phyla, and we cannot really assign them any function. And so this is the problem. And this is so we have this. So how do we tackle? So so now we know we have these things. They are being conserved. So there's some biological process happening there for evolution to preserve them, but we don't really know what they're up to. And so this is now what we're trying to figure out. And the solution that we arrived at is to use, and we were not the first ones to do this, but the solution we arrived at was to use machine learning. So we took all of these small open reading frames, and then we developed a tool called microbial, which I'll present in a second, to basically tell us, is this an antimicrobial peptide or no? So an antimicrobial peptide is, as the name says, it's a peptide. So in our so a small protein that's antimicrobial, meaning it stops stops bacteria from growing, or it just outright kills them. And so we took we took our tool called microbial. We developed specifically for this project. And so the way microbial works is, so we take these, we take our metagenomes, we do this assembly, predict the genes, and then so this is sort of old-fashioned machine learning. where we are computing some features, and I'll show you in a second. And the reason why we do this, so I said before that homology is difficult, and we did try to benchmark it. So here in this is the accuracy we get with using homology. So in a homology is the blue line, so blast, and then using a couple of different tools. So Macrel is our tool, AMP scanner and IAMP2L are different tools, and so the couple of things I want to show here. So first of all is that there's a huge gap between what you can get with homology and what you can get with machine learning based tools. So the homology based tools are basically useless, because again, unless you have very, very close homologs, so if you have two sequences that are 90% identical, then okay, that's gonna work. You're gonna be able to do this functional transfer of, okay, if this sequence has been annotated and then this other sequence is 90% identical to it, then okay, we can do it. But it's gonna very quickly lose ability to do that. The other thing I want you to notice is that if you look at our tool Macrel, it's actually not the best in terms of accuracy, and it actually has pretty low sensitivity, but it's the best in terms of precision, and I'll get back to that in a second. Yeah, so as I said, this is a standard machine learning sort of old-fashioned machine learning. Is that so? We take a sequence and then we compute these features that some of them were previously published, others we designed. Where so for example, also just to give you an example, here in C we have this sequence VTSWS. So this would be a peptide, 24 residues that we're looking at. And we group the residues in groups, and then we see how they are distributed within the peptide, right? So are they do we see these groups at the start versus the end of the peptide, and we fit this into a random forest, right? And the one thing that we also do is we train this random forest with a data set, and that I'm showing you here on the left, that is mostly composed of non-AMPs. So it's mostly composed of the negative class, about 98% to 2% of the positive class. And I'm going to compare this with this alternative method, which we only use for benchmarking, but let's call it Macrel X, which uses a balanced data set as training. Right, and why are we doing this? And maybe for many people here, this is just very basic probability theory, but this maybe allow me just to recap. Also, I'll do a little bit of the numbers. I think it's also good to see the numbers, right? But so this is the standard, so the standard Bayes rules, right? The probability that something is an AMP, if the classifier says it's an AMP, it's the probability that the classifier says it's an AMP, given that is an AMP, of times the ratio of is AMP over the classifier says AMP. So this is Bayes' rule, right? And now if we expand this right sign, the probability that something is AMP, it's basically depends on how good your classifier is, and there's how sensitive the classifier is, and how specific your classifier is, but also in the underlying probability distribution that some peptide is or is not an AMP. Right? And let's just give some numbers to this and see what comes out. So let's say you have this classifier and it's 95% specific, 95% sensitive, but the base rate is only 1%. Right? Because in real life, when we look at the metagenome, we expect that very few of our peptides are actually going to kill bacteria. So most of the peptides are going to be anywhere from false positives. And there's been small peptides that have been reported to be involved in all sorts of different cellular processes. They're involved in gene regulation. They're involved in in membrane biosynthesis, they're involved in cell communication, they're involved in all sorts of things that are not killing other bacteria. So the base rate, let's just put 1% as sort of probably not exact, but probably in the right ballpark. It's closer to 1% than 50. So if we have these numbers, then if we have something, if the classifier says that something is an AMP, then if we work it out, then actually the probability that it's really an AMP, it's only 16%. Just because the base rate is so low, because the base rate is so low that it goes from 1% to 16, but it's still you're still going to be almost always wrong. So in fact, the specificity matters a lot more. So we are going to be willing to sacrifice a lot of sensitivity if we get higher specificity. So for example, if we get the specificity all the way to 99, even at the cost of sensitivity, then now we will be closer to 50-50. But even with 99% specificity, we're still at 50-50. And if we take the specificity all the way to 99.8, and I'll show you, then now the probability that something really is a true positive, now it's in about the 80% region, right? So this is why we felt we had to develop our own tool, because we wanted something that was really, really tuned for very high specificity, even at the cost of sensitivity, because we have so many data nowadays, we have so many data sets that it's okay if we miss something. We're perfectly fine. If we miss something, we have billions of candidates, but we want to be very confident that when we call something an AMP, it really is an AMP, or as best as we can, right? So I was playing around with these numbers, but 99.8 is actually what we got in our benchmark. So here we have a couple of different tools that were tested at the time, actually. So Macrel came out a few years ago, so there's been even more tools that have come out. But I think this is still true that we don't do the best in terms of overall accuracy. We don't do very poorly, but we're not the best. But when it comes to specificity, we do get this 99.8%, which is significantly higher than other tools, and it's significantly higher than tools that have higher accuracy on these benchmarks, because these benchmarks don't have a realistic balance between true and false positives. Right. So okay. So right. So and here's Macrel X. So this version, that's exactly the same architecture. It's just being trained with a balanced data set, and it does better in terms of accuracy, but it has lower specificity. And as I said, specificity is what we're really trying to optimize for specificity. So we're willing to sacrifice some recall as long as we are as accurate as possible. Right. So we did this validation, and we've published a tool. So this is also available as a tool. It's available. We have extensive documentation. So we have YouTube tutorials, and we really try to be responsive to queries and bug reports. So we have this commitment, and to high quality software. So and we want even the people out there who maybe don't care anything about what I was talking about and the machine learning and the statistical stuff. They really just want to have good predictions for their data. We want them to be able to use it easily, even without a lot of, even if they don't have all of this bioinformatics training. All right. So going back to but we had this tool, and now we applied it to this big data set. And we generated what we're calling the AMP sphere. So the AMP sphere being the set of all of potential AMPs from this global microbiome. And so DRUMP is an existing database of AMPs, and green is so this is just sort of the length most of our antimicrobial peptides are in this range of about 25 to 45, 50 amino acids, right? And they are being produced by all sorts of different bacterium, although it ends up reflecting what are the organisms that we have in our database. So we have a lot of human gut data. Prevotella is a very common human gut bacterium, and so we end up with a lot of sequences that come from Prevotella, and so and Bacteroides is another human gut bacteria. So this, to some extent, a lot of this is a reflection of what the data set is. So if we have a lot of the but one way we can correct for this is to say, okay, if we have certain amount of bacteria sequence coming from Prevotella, we expect to see more AMPs coming from Prevotella. So let's divide one by the other, and we're going to call that the density. So the density is, if we have 1 million base pairs of Prevotella sequence, how many AMPs do we see? And we define this density, and so in general, the density is going to be it's going to be about maybe a half to 1, something like that. And we then took another paper. There was a paper from Nicolas Segata's group, Falis Komar et al., that came out more or less as we were wrapping up this manuscript. And they asked a completely different question. So they asked, what determines which bacteria are easily transmitted between a mother and their offspring, or within a household, or within a population. So we have our gut bacteria, and if we happen to be roommates, which kind of bacteria are more likely to be transmitted? Because I think at some point we used to think that there was a general answer to this question, and it turns out that it's very, very specific. Some species are more transmissible than others, and we took their data, and then we took our estimates of the AMP density, and we correlated one versus the other, and there seems to be in this close contact, so mother to offspring, we saw that there's a correlation between the AMP density of a species and how transmissible they are. So things that are more produce more AMPs are actually less transmissible. Maybe because the AMPs are actually protective against transmission. So these things are often competitive. But all right, so far everything I've shown you was, we were doing computational work. We developed tools. We benchmarked our tools on existing databases and existing benchmarks, and we applied them to this large data set. And then, but does it really work, right? And then we teamed up with Cesar Lafuente's group, and Marcel Torres is a postdoc at this is at the University of Pennsylvania. And so then we took 100 candidates, including 50 that were completely at random. So and one of the advantages of these small proteins is that they are very difficult to work with computationally, because of all of the statistical elements that I mentioned before. But they do have one big advantage in the lab, which is you can synthesize them much more easily than you can the longer proteins. So we can synthesize them, and then we can test them. We can see whether, when you give this to bacteria in culture, does it really work? Right? That ideally, you give the peptide and the bacteria stops growing. So Marcelo took 2 groups of bacteria. So on the one hand, these really bad guys, so these escape pathogens. So these are the pathogens that are often antibiotic resistance. They're the ones that are really, responsible for a lot of deaths, unfortunately. And then he took this other group, which are the commensals. So these are, you know, the things that probably most of us have in our guts that, you know, seem to be at worst neutral, many of them even probably beneficial. And so so the first question was, you know, does it work? And so so here I look, so in this heat map on the right, so if you see a square that's darker, that means that that's a hit. So that means that that peptide inhibited that bacterium, and it's probably a bit difficult to count, but in the end, out of 179 were active, right? And obviously it's hard to prove a negative, so it maybe the other ones were not active because we didn't test them right. We didn't test, but it's still actually quite interesting that, you know, so before I showed you that if we do this very basic Bayesian type style analysis, and we put in some plausible numbers, and we say, what if we have a tool that's 99.8, which is what we measured for Macrel in our benchmark, in terms of specificity. You'd expect that you'd get about 80% true positives in your data, and then we got more or less exactly that. Right. Okay. I'll take the questions at the end if they're more generic. But so I saw there was a question in the chat. So if it's very specific to a slide I'm presenting, I'll address it. Otherwise, I'll just wait until the end. So this is also maybe this as the human element to this as well, is that we were working for years on this project. Right, we published the tool as a milestone. That was great, but our goal was always to do this survey. So we do all of this computational work. We're talking, months of just stuff running on the cluster and all of the predictions, all of these discussions on exactly how to do it. Then Marcelo goes in the lab, spends, all of, you have to grow these things, so there's years of work and sweat and labor. And then it's only right when you finally look at the data, it's like, there's a moment there where it's like, you're not sure if it's going to work or not. And whether all of these theories really, that's the moment of truth. But for us, it really does seem to actually work pretty much as we'd expected, or as we had hoped. And so we also took then some of these top candidates. So here you need to take a few other things into account, not just whether they work in vitro, but also whether their peptides are stable. And then we took them into a mouse model. So this is a skin infection model. So right, imagine you have a cut on your hand, and you have an infection. So you can apply, basically you apply cream, an antibiotic cream, locally, and here we use, so in the gray we use polymyxin B. So this is a positive control. This is actually an antibiotic that is used in exactly this situation. So for certain infections, if you go to the doctor right now, you might get polymyxin B applied as a cream. And so this is our positive control. And then we can see that some of our peptides are actually very, very close, and actually even statistically indistinguishable from the positive control in terms of this is, there's still room for optimizing these peptides further, but this is very close. This is very close to something that would be usable. Right. Maybe I'll give you one or two more stories that we have from this data. So I've been using this terminology AMPs, and but actually, so often in real life, including the AMPs that we produce as humans. So humans also produce antimicrobial peptides, and the ones we produce are often we start with a big protein, and then there's a protein cleavage. So and a little chunk of that protein cleavage gets exported and is now the active peptide. Right? And some people would say that actually that's what you should, you should use the word peptide when you started with a large thing that was cleaved, and that's but whereas we were looking at things that are their own gene in the genome, so this is a particular subset. But then we asked, but maybe they were cleaved not as a protein that was cleaved, but as a gene that was cleaved. So this was some sort of genomic equivalent where, and so we explored this hypothesis by taking our predictions and aligning them back to this catalog that I showed you at the very beginning of full-length proteins. And so I'll caution that we can only get matches for 8% of the whole data set. So this story does not, it's one story, it's one origin story, but it's only maybe explains this one fraction, one part of the data. But what was interesting for us is that so of the ones where we find the match, about 1 in 4, the match has exactly the same start codon, and then somehow there's an interruption. And I'm just going to illustrate that with an example. So in this example, so we have here the green. So this is a real long protein for NADP dependent dehydrogenase from Prevotella jejuni. And then at the bottom in the purple, there's the same protein from Prevotella melanogenica. And then in this orange, we have our metagenomic context. And so here in bold, there's the ATG. So that's the start codon. There's a ribosomal binding site right a little bit upstream. And the sequences align almost perfectly. There's a couple of mutations. And then eventually there's one mutation where there's a G that turns into an A. And so that produces a stop codon there, right? And the dehydrogenase keeps going. But with that stop codon, now we have a fragment, like we have a smaller protein. And so, again, this is very hard to actually prove this, but our model of what happens is that, this type of stuff happens all the time, right? You have a mutation, now you have an interrupted protein. And almost always, this will get, evolution will take care of removing this from the genome, because now you have, but every so often, maybe evolution gets lucky, and now this smaller fragment is actually active. And now this smaller fragment, you've generated a new protein, which, no longer, it doesn't even need to have any relation to the original one, but because now you have this. So maybe this is one way in which these things get generated, is that it starts as a long protein. Every so often, evolution gets lucky. And actually, what we sometimes see is that we see these fragments, and they happen to be right next in the genome, or at least very close to the full protein. So here, so here in this representation, so these gray boxes are these AMP 15426 that happens to be homologous to RPSH, which is a ribosomal protein. And we can see in these genomes, often it's right in the middle of other ribosomal proteins, and often right next to the RPSH full-length protein. So here again, it's very hard to prove what happened historically, but one model is that there's this partial duplication. The partial duplication is interrupted. But now the organism retains both the original protein that's probably still doing its original function, and now it's gained this small variant that if it gets lucky, this small variant has its own function. So this is one origin story for how these peptides come about. Right. So everything's available online. So there's the website. There's there mpsphere.bigdatabiology.org. You can just search for things. You can just browse and click around. Everything. There's an API that you can use, or you can just download everything yourself if you want to do a lot of processing. There's actually a lot more that I didn't talk about today, just because of interest of time. So we have, you know, there's a lot more details in the paper, including some more data on exactly how the peptides work. They disrupt membranes. We can see that they disrupt membranes. And so just to summarize, so these small proteins, you know, there's billions of them outside. We know that they're really conserved. And they're conserved both in terms of evolution, in that we see them in different phyla, and they're conserved in terms of ecology, that we see them in different habitats. So, you know, they're doing something, but we really don't know what they are. And so we started with, so here, you know, I wrote a focus on antimicrobial peptides, but for us, this is the start, is that, you know, because they're important, but we are really trying to understand. We're really trying to understand, you know, how could we approach, you know, these billions of sequences. We know that homology doesn't really work. There's a lot of limitations on homology, and so for us, antimicrobial peptides, we decided to tackle it with machine learning, and this is what we're exploring. And then, you know, we worked with Cesar and Marcelo, and we were really, you know, we've got this in vitro validation. So with that, I just, you know, most of the work I showed was from Celio and Ichian and my group, Marcelo and Cesar's group. But then there was also people in Heidelberg, where, you know, who did a lot of the work in making sure that the datasets were, Alvaro and Jaime in Madrid. So this was funded. So this project started when I was still in Shanghai. So we had funding from the National Science Foundation of China. And now I'm in Australia. I'm funded by the Australian Research Council, who gave me a future fellowship for this work. So I want to thank them. And now I want to thank you for your attention. And I'll also post, so I'll post this link in the chat as well about so we have, if you want to find out more. But I'm going to go over the questions I see in the chat as well. Unless someone else wants to step in. So I'm going to read the questions aloud. So good morning. Sarah, can you say how you select peptides, if it is for specific diseases, for chronic kidney disease computation? Can you suggest any database? So this is actually a really good question. So there's a lot of research on peptides more generally for all sorts of different functions. So the antimicrobial aspects, especially antibacterial. So I've been a little bit imprecise with that. So especially antibacterial, that it's really been much more studied. So there's been more databases for it. I mentioned, where is it? I mentioned DRUMP at some point. So DRUMP is one database of antimicrobial peptides. But there's other functions that, so I mentioned the DRUMP, but there's other functions in particular, anti-cancer, etc., that are being really explored now. But it's still there's still much more research to be done. Thank you. It is really a fascinating piece of work. Thank you. How the peptides did you isolate it from the source or chemically synthesized? So these were chemically. So this is a good question. So these were all. So for our data, they were all chemically synthesized. So I think if you want small amounts of peptide, this is a very good way of. I mean, we worked with Cesar. They're very good and they have a lot of experience, especially especially these are relatively simple peptides. So this was all chemical synthesis. This question about the diversity of protein structures. That's a really nice question. So again, there's just not a lot of amino acids. So many of them, the structures are relatively simple. So often you have, for example, a helix and then a loop. That's a very common structure. But you just don't have enough, a lot of, like if you have something with with 30 amino acids, there's just not a lot. You don't have enough amino acids to build a big, 3D structure. So they're often also, you don't have a lot of amino acids to keep that structure in place. So sometimes you have these flexible areas, or you might have a helix, but that helix sort of is not very stable. But often, yes, helix and loop, or a beta sheet and loop, where the beta sheet is also, goes there and back, right? There's just not a lot of amino acids, but I would like, so we haven't really done any of that work here, but I would like in the near future to, if possible, be able to look a lot more at structure, and look a lot more at some of these questions. For how much of a amount of peptide is toxic, or can we take it in vivo? It really depends, actually. So this doesn't really have a, so in our case, we actually don't know. So in the mice study, so we know that the mice didn't lose weight. So it means it's not very toxic. But none of these sequences are sequences that you should, if you're sick, you don't go and synthesize them and take them tomorrow, right? So it's really dependent. It really depends. There's right, we consume grams and kilos of protein in our diets, and that's fine. And then there's other things that would be much more toxic. So there's really no answer for that question. But checking toxicity would definitely be would definitely be a step towards taking this forward. AMPs can attack every layer membrane because of their positive charge. Is there any so again, I mean, I hate sort of repeating this answer, but yeah, there's a lot of, so maybe this isn't, that there's a lot more that we still don't understand. So let me just put this. So some of these peptides, right? Or none of these peptides kills every bacterium, and sometimes the effects are very specific to. So we don't get what we would like to get is and actually maybe I'm going to answer the next question already. So the next question was, how did you choose the organisms to test these against? So to some extent, on the pathogens, it's a little bit easier because to choose, in the sense that so there's these escape pathogens. At least if we're worried about human health, so these are these have been designated by including the World Health Organization, but generally, there's a big consensus that these are the really bad guys. Like these are the ones that really so if you see right and so especially in terms of and so there's so this is so ideally we would get the peptide that kills all of them and then doesn't harm the commensals. So choosing the commensals is a little bit more difficult in the sense that ideally we would choose a lot more. There's some, and here we had to make a little bit of a judgment call in terms of trying to get a couple of different different taxa, not just testing. Yeah, not just testing. Yeah, this was a bit of a judgment call of trying to get the most things that are very abundant in the human gut, very prevalent, and then we ended up with these Akkermansia, Bacteroides, and etc. But we see these. So the point I wanted to make is often we see these specific effects, and we don't really note how to explain why these effects are so specific. We don't know how to explain why is this bacterium targeted and not that one. So this is another one of these things that are that we would like to understand better in the future. I think and especially being able to predict, ideally we would say, okay, we want something that targets Klebsiella pneumonia and only Klebsiella pneumonia, right? One of the problems with standard antibiotics is that they might kill Klebsiella pneumonia, but then they also target our healthy microbiomes, which has other deleterious side effects, which also helps with spreading antibiotic resistance. So having this more targeted, so we think it would, we think we could do better there, but we still, we don't know how to do it. All right, so, okay, let's see. There may be community difference between healthy and diseased person. How for AMPs used in precision medicine? Can we validate the results in silico for in vitro? So how can you comment on this? Okay, I mean, I think I was trying to get a little bit. I was trying to get a little bit at this. Yeah, so being able to say, okay, like you go, this, again, this would be something that we're aiming towards in the future. If you have, you can go to the, you go to the clinic and you have an infection, and you get this rapid diagnostic says, you have Acinetobacter baumannii, and so if you have Acinetobacter baumannii, then this one particular peptide would be the best for you because it's going to target Acinetobacter baumannii, but none of the other ones, and it's going to leave your gut commensals alone. So this would be the ideal. So right now, as I said, this is all empirical data. We don't have a way, we don't really have a way of being able to do this more in silico, in particular optimizing this in silico. And I think I think this also answers the next question about what factors contribute to differences in efficacies amongst. We really don't know. So there's been a lot of work on specific peptides, especially the peptides that are already being used in the clinic. So actually, using peptides as an antibiotics actually predates penicillin. So this was used already in the pre-penicillin days. I mean, obviously at the time people didn't know it was a peptide, right? But this was the, and it didn't really work as well as penicillin, but it did. So we would like to be able to, understand, like predict. Okay, if you have this peptide. So sorry, what I mean is like, so people have studied very specific peptides and say, okay, this is, and being able to, and they've been able to understand that certain peptides are affected by certain lipids in the membrane, for example. But this is always done after the fact. So we're not able to go from. So we have not been able to go from. This is the bacterium that's infecting this individual. Which peptide should they take? This is all done so far. This has all been done empirically, including our own work. This is all empirical. This is we haven't been able to predict yet. So, but this is the work. This is the work we'll try to do in the next few years. Oh, can you? Oh yeah, this. Okay, and okay. Is that? All their novel peptides acted on bacterial membranes. So, yes, can we use AML to identify intracellular acting AMPs as well? I mean, in principle, yes. I'm not sure if we have the data sets for that. So I'm gonna. So I mean, a lot of this depends on the data sets that we have, right? We're building on training data. But in principle, yes. The question is whether we have the training data for that. All right. If we got, yeah, okay, yes. I mean, I think if we encounter antimicrobial resistance, is there a way to understand how normal peptides would interact with them using machine learning? Yeah. So the peptides are, yeah. So I don't, again, so these peptides that are antimicrobial are a small fraction, right? Most peptides, I mean, when we eat any protein, be it, animal protein or plant protein, like we're ingesting peptides all the time, right? We're ingesting, and 99.9, when we're in contact with anything, right? So most of the time, these things are not going to be particularly active either way. So someone was asking about the slides. So the So the slides are just publicly available, and generally and including the slide with contact information. So, feel free to contact me afterwards as well. I could, okay, yeah, sorry. So I'm, one of the most concerning aspects of antimicrobial resistance is the ability to create biofilms. Is there any server software to precisely simulate biofilm and analyze peptides biofilm activity? Except DBAAP that analyzes peptide protein. So actually, as far as I know, so this question about biofilms is a very good one. As far as I know, there's no software that simulates these, as far as I know. But that could just be my ignorance. Yeah, actually, yes, yeah, it's a very good question. So this is maybe a limitation of all of these studies, the in vitro studies. These are things growing in suspension. In a biofilm, it could be different. When we go to a mouse study, maybe that partly also explains why some things look good here, and then in the mouse study, some of them, basically, obviously we talk about the positive results, but there are also some negative results. So here, so we have the positive control, but we also have the negative control, and a few of them, basically, they behave as the negative control. So maybe that also partly these more complicated structures of bacteria explain why. But again, this is all empirical data. We haven't, ideally in a few years we would want to be able to say, oh, this one is not gonna work because of biofilms, and that one is gonna work well despite the cells being in the biofilm. All right, I think I've gone through. I mean, and we are well over time. I don't know. Maybe I should give over the meeting.
Thank you, Dr. Curlo, for this informative and engaging talk. And I would like to extend my sincere thanks for taking your time to share your insights.
Oh, thank you. Thank you for having me.
Your talk was incredibly informative, and it left a lasting impression on the audience, I'm sure, because you got so many questions and you answered them with real patience. So I would like to thank the participants as well for asking the questions. So yeah, for people if they Dr. Coelho, and I would like to tell that there is a survey that you can give your feedback on, and the link for that is posted in the group chat. And also, the whole session was recorded, so you can, if you want to go through it again, then you can go to YouTube channel of the AP Bionet Talk, and then the link for the same the YouTube channel is also available in the chat. So I would like to finally thank you for taking your time, and I would also like to thank the organizers of AV Bionet for making me a part of this. Thank you.
Thank you everyone for joining. We hope to see you again on the next event. Thank you.
Thank you.